Proteins that are destined for the lysosome are synthesized in the ER, like all proteins.
Targeting proteins to the lysosome begins in the Golgi. More specifically, this is the trans-Golgi network. The Golgi is where addressing molecules bind to the cargo.
In the trans-Golgi network, the sorting signal, mannose-6-phosphate, M6P, becomes attached to a damaged protein.
The M6P signal will then attach to an M6P receptor. This will be useful for attaching clathrin to create a vesicle. The vesicle is demonstrated by the trailer.
Clathrin cannot bind directly to the vesicle. It requires an adapter complex, AP1 in this case, which is represented by the hitch of the four-wheeler.
GGA, represented by Martin himself, is the protein that attaches all of this together.
Rab9 directs proteins out of the Golgi, like the handlebars of the four-wheeler. Clathrin forms a vesicle and transports it to the lysosome by the same process discussed in class. That is, budding, fission, uncoating, tethering, docking, and fusion.
The M6P receptor is then recycled.
Lysosomal enzymes are transported in pretty much the same way. M6P binds, then the M6P receptor, clathrin, and AP1.
A clathrin-coated vesicle then forms and is transported, this time to a late endosome instead of a lysosome.
Using these enzymes, a late endosome matures into a lysosome.
The lysosome then degrades the protein.
In the arms of the angel, fly away from here.
But that's just the major pathway. There is also a minor pathway where the vesicle exits the cell.
At first, there are no coat proteins. The vesicle buds off of the Golgi with the cargo, M6P, and its receptor.
The vesicle then travels to the cell membrane where it fuses. Endocytosis occurs using clathrin, and then the vesicle is targeted to either the lysosome or late endosome.